to the cloud. All right. So today we are going to talk about metabolism. And I do like to start with a picture of my head so that I don't get in trouble copywriting things, right? But, and not that first picture of the PowerPoint of the thing from the textbook. So today's chapter 8, today is metabolism. This also, like chapter 5 and chapter 2, should be a quick review for you guys. You should be pretty fluent with this by now. Most of you, I believe, have taken Chem 1 and 2, and this is a repeat of a lot of the things that you would have learned in Chem 2. So I'm going to go through this fairly quickly. The PowerPoints, I just put them up there. I would use these as your note outline. Because I specifically hit the things that you have to know for the exam. That first exam, remember, is coming a week from yesterday. And I know I promised that I would put up more Connect assignments, and I didn't. I promise that I will do them today. And you guys just need chapters 2, 3, 4, 5, and just a little bit of 5, really, 5 and 8. So I feel a little guilty, but it's still more than, you still have more days before the next exam than you will have assignments. So I figure if you can do them one of them a day, it'll actually really help you get ready for the test, as opposed to if you did them 3 weeks ago, you might not remember the material. So my apologies, 100 apologies. Like you, still doing the best I can, right? With the equipment and the time and all that stuff. So that aside, let's run through this metabolism stuff quickly here. So just definitions, overall definition of metabolism, sum of all the chemical reactions in the cell. We can divide that into catabolic reactions or catabolism, which are the breaking down reactions. Those usually release ATP. Anabolic reactions are endergonic, usually require ATP. We are skipping photosynthesis. So even though it will appear at the end of your chapter, you can just bleep, don't even look at it, okay? We're not gonna go over that portion of it. We're just gonna focus on the catabolic processes here. All of these pathways are controlled by enzymes, right? The pathways that we're gonna focus on, glycolysis, Krebs cycle, and the electron transport chain, right? I don't know what to skip. I should have just deleted that one. Quick review of enzymes. They are globular proteins. I'll draw a picture of one for you on the whiteboard in a second. They end in ASE. That's how you know if something's an enzyme. Remember, the protein portion usually requires a cofactor or a coenzyme, this should say, right? A cofactor is usually a metal ion like zinc, aluminum, something like that. And coenzymes are organic, non-protein molecules. Think vitamins, usually, but that's a coenzyme. Structurally, enzymes have an active site. That's where the chemistry takes place. The substrate is the molecule that fits into that active site. Remember, that's either going to be joining together. What's going to happen in that active site? We're going to form a chemical bond, or we're going to break a chemical bond. Not on my list here, but should be, right? So they lower the activation energy. They are similar to catalysts, right? But except they're not used up. We can use them again and again. And the other thing that is not on here that should be, I think, is that enzymes are super specific. Usually you have one enzyme that performs one chemical reaction. They're very specific. This is why we need these long pathways in order to, that's why we need glycolysis has 8 or 10 steps, because each enzyme in the pathway can only do one thing. It can break a bond between this carbon and this oxygen, and that's it. And that molecule has to go to the next enzyme to add a hydrogen to each of those, or whatever the next step is. So that's why they're in pathways, because they're super specific. That, again, that should be on this slide, and it is not. And this is sort of generally what all the rest of what we're going to cover about enzymes, a little bit about the regulation. I'm not showing you that picture. I just said all that. All right, so let's, I'm going to stop this share for a minute, and I'm going to draw a few things on the board. I don't know why that did that. There we go. Make me big. There we go. not on there. Running over here. And I have an eraser somewhere. Where's that going? Okay. So clearly we were talking about the size of cells in Bio 1 today. Okay, see how big that's board. So what I briefly wanted to say about enzymes as far as the structure of an enzyme is the following. I usually would draw an enzyme on the board. Remind you, they're made of polypeptides, so it's chains of amino acids that are folded into tertiary structure at minimum, quaternary structure for some enzymes, meaning they have subunits. So if I were to draw an enzyme, I usually kind of put some beta-pleated sheets and some alpha helix, right? And sorry, I'm going to make that a little smaller here. Actually, I'm going to bury that inside. This would be the ammonia end or the amine group. So I'm going to put H3N here for that first amino acid and draw my carboxyl group here, COO with a minus for my last amino acid in the chain, right? Oh, wait, I think I have some colors that are functional. Let's do the purple, the green shows up pretty well, I think. So if I would have a cofactor or a coenzyme, let's say I'm just going to put a square, kind of a square shape here, and I'm going to label that ZN for zinc. Well, I think it's a +2. Don't quote me on that, right? So that would be an enzyme with a cofactor. This is a key to help this enzyme turn into this particular structure. So it's absolutely necessary for the structure. If I take that zinc away, the enzyme may not be completely denatured, but it certainly won't form the exact shape that it needs to form. Since they're proteins, I would remind you that they can be denatured by heat or changes in pH, right? And then, of course, this area here that I'm going to highlight in yellow, and I don't know how well it'll show up on your screens, but I'm going to highlight it in yellow. That's the active site. So that's where the substrate is going to fit. So I'll make the substrate this sort of bluish color here like this. All right, so the substrate has to at least have this shape in order to substrate to fit into the active site, just like that. So the place is the active site, and the molecule that fits in is the substrate. This can be one thing, like I've drawn it, or it could be two things. If it's an enzyme that builds, we might have two substrates that fit into that active site. Reminder from Bio 1, this substrate has to be the correct size, the correct shape, which I've tried to highlight here, and as well as the correct charges to fit into the active site. Okay, so that yellow part, remember, is the active site. That's where the chemistry takes place. That's where the chemical bond is going to be made or broken. Right? What do enzymes do functionally? Over here. Right? We've said already, and I've shown you on those PowerPoints, right? They lower the activation energy required for the reaction to take place. And what we mean by that is it lowers the activation energy, and what that does is it increases the speed at which the reaction can occur. And because it is a biological catalyst, it is reusable. That's really important. So it speeds up the reaction, and the cell can use them over and over again. They're proteins. They're really expensive to make. So once the cell makes one, or 50 of them, right, it's going to need to use them over and over again, or it's not going to be very efficient for the cell. Okay, All enzymes end in ASE, we mentioned that. And the reason for the specificity is because of this size, shape, and charge of the substrate, right? Only this substrate is going to fit into this particular enzyme. Okay, now I apologize, but I'm going to bring you back over here so we can go through some more of those PowerPoints. Carrying around, so I can leave my box set up over there. Maybe. Okay, so let me go back to sharing the screen with you for these PowerPoints. Right, I'll make myself go away completely. Don't need to see me. All right, so we did this, right? This is your book's rendition of what an enzyme looks like when the substrate is fit into it. You have a smaller version of that in the current edition of the textbook. Notice they're trying to point out that the shape is important as well as the size and the charge, and then here are the products that come out, right? Proteins are always purple, just like in your Bio 1 book. So enzymes are proteins, that's why they look like this. Now, here's just a list. I like this table, and I always include it if possible, because it shows you how their names all have ASE at the end, almost all of them, and that their names tell you what they do, right? So an enzyme called lactase, what it does is it breaks down lactose. So a lot of times, if you know a little bit about organic chemistry or a tiny bit about the names of the molecules, you're able to very quickly figure out at least part of the enzyme's function. So that's kind of a useful tip for later, right? So regulation, metabolic pathways, skip. Genetic control, that really isn't a thing. So I'm just gonna run through real quickly with you. I'm gonna stop this share and run through real quickly with you mainly the ways that enzymes are regulated, which I believe your textbook would refer to them as metabolic regulation of enzymes. So let's just do this. This dog is lining these shirts. Okay. All right, so I'm going to erase this, even though I would like to keep my beautiful enzyme up there for a little bit longer. I'll try not to write in this white spot, but I think it's the computer trying to give us enough light in order to record the webcam. All right, so regulation of enzymes, just again, real quickly, you can review this stuff in your Bio 1 notes. So regulation of enzymes, and we're talking here about environmental regulation, right? So meaning inside a cell or inside a body, in the case of microbiology, inside the cell, or maybe inside a pool of water, a pond of water, or the soil. So the regulation is by temperature, pH are the big ones, presence or amount, AMT for amount of substrate. And then I'm going to put down at the bottom here inhibitors. Okay, that's just to help me remember all of them and to hit all of the high points for them. So for temperature, the enzymes that we're talking about or we're mostly going to encounter work inside cells that are inside humans, and our temperature is 37 degrees Celsius. So those enzymes are going to work best between, we say, about 35 and 42 degrees Celsius. That's when they're at their optimum. And if you heat these enzymes to a temperature, let's say I would say over about 50 degrees C, they denature. Basically, you cook them, their chemical bonds get broken over here, and they no longer have this shape. And if they no longer have this shape, they no longer have an active site that the substrate can fit into. That's what denature means, shape changing. Cold is not as effective. Okay, so low temperatures are going to slow enzymes down, but they are not low, right? Low temperatures will slow them, but they will not necessarily completely inactivate them. Freezing, maybe, but you have to freeze at literally at like minus 20 degrees Celsius if it's an organism that can cause disease in humans. that's why we use freezers and refrigerators to help preserve our food, because it will slow down or completely inhibit enzymes. But unless you actually denature the enzyme, when the whatever it is reaches back to room temperature, or if it's a body temperature, some bacteria, those enzymes will start working again and could still cause disease or sickness. pH, I would say acids also denature enzymes. So acids denature, particularly low acids. pH is 1, 2, 3, 4-ish, 5-ish maybe. Stomach acid is a little over 1, right? So acids will denature and cause enzymes to be inactivated. Bases as well, but not quite as much. Now, why in cells does this not usually affect enzyme activity? Because remember, inside a cell, cells have buffers to help moderate their exposure to acids and bases. But if I have a bacteria or a group of bacteria and I put them into vinegar, right, or I put them into a stronger acid like lemon juice, that is going to denature the bacteria. That's why those things are preservatives or can be used as preservatives, right? Many of the acids that you find, like even when you read the label on a loaf of bread, many of those acids in there, or any of those words that end in acids, they're there as preservatives. And what that means is they're not artificially changing the bread that much. What they're doing is they're inhibiting the enzymes from the bacteria and mold that get on that bread as soon as you open the bag the first time. So they're helping that bread be able to last you for a week, right? Amount of substrate, that's pretty self-explanatory. If substrate is present, the enzyme is going to work at optimal pace. If there's no substrate or it's a low concentration, the enzyme isn't going to be working as much. And then inhibitors, we should address real briefly. They come in two forms, competitive and non-competitive. And they are just like they sound. The competitive inhibitors compete with the substrate for the active site. The non-competitive inhibitors, they bind someplace else, and they change the shape of the active site. A lot of pharmaceuticals, draw that in green, I guess, are non-competitive irreversible inhibitors. So they bind to a particular enzyme that may only be in the bacteria and not in the host, aka you, and they inhibit that enzyme permanently. So they bind out here somewhere, and they change the shape of the active site, and it's a permanent binding so that that enzyme can never digest that substrate again, and will eventually kill the bacteria. Right? So that those non-competitive inhibitors will return. And actually, some of the sulfa drugs are competitive inhibitors. That'll return to us in a later chapter. So we'll see those again. All right. Now, let me go back to these PowerPoints here and skip along through here. Enzyme characteristics. Whoa, there's a nice long table for you to read about. Now that we've talked about enzymes, which are the molecules that regulate all these pathways of metabolism, now we can talk a little bit about energy and how these enzymes actually produce usable energy for cells. So I would remind you, ATP is the form of energy that's used by cells, and that energy is created by oxidation and reduction reactions. I already mentioned these words. Exergonic, remember those are usually catabolic reactions. They release ATP, and endergonic usually require an input of ATP. We're going to take a look and talk about redox reactions and electron carriers a little bit, again, as well as the structure of ATP, since that's the actual molecule that cells use to do their work. So this is just a reminder, but a poor one, I think. Electron carrier, yeah. Okay, so we're missing a little bit of data here for the redox reactions. Again, I'll just write that on the board rather than type it on the PowerPoint, because I don't want to change too dramatically the PowerPoints that I've already saved and loaded on your page. right? So I want them to be relatively the same as these. I still can't erase that picture. Okay, you can do it, right? You can do it. I need it for anything else. No, I'm gonna erase it. Okay, so. Redox reactions. RXN for reaction, right? Redox or oxidation reduction reactions. We've learned about them in chemistry. The molecule that is oxidized. So the molecule that's oxidized, I'll give you a minute here, gains or loses electrons. Think about it. Compared to the molecule that's reduced. I remember it differently than what my children told me about this little device to help remember that. Oxidation involves loss, reduction involves gain. Or my other child told me, no, we learned Leoger, L-E-O-G-E-R, loss of electrons oxidized, gain of electrons reduced. So either one of those, we call those, right, mnemonic devices to help us remember. Either one of those I would use. And I also tell students I would always only remember one of them and then just puzzle out the second one. So myself, I always remember that reduction is the opposite of what I think, right? So reduction, I would think, means loss of electrons, but it's actually gain of electrons. And the molecule that's oxidized, that's the one that loses the electrons. So loss of electrons. You guys are okay with me writing this, right? E with a minus sign for electron. Now, this is the part that often gets lost in the discussions and descriptions of this in Bio 1 and other courses. You can't do that in a cell. You can't just take electrons and move them off of one molecule or atom and stick them on another one. In particular, you can't do that to oxygen because that makes it become super electronegative and it causes mutations and death to the cell, or mutations and cancer if it's an organism. So in cells, I always try to emphasize to students, inside cells, you can't just move electrons around. You have to do it in a safe way. And the safe way to move electrons inside cells is to move hydrogen atoms. So we still have oxidation, but instead of a loss of an electron, it's the loss of a hydrogen atom. Because hydrogen atoms, you might remember from somewhere in your past, hydrogen has one electron and one proton, right? And 0 neutrons. So they're very small and easy to move around inside the cell. The molecules that are reduced are going to be not gaining just electrons, they're going to be gaining hydrogen atoms. Atom or atoms. I say atoms because these hydrogen atoms, we're usually going to be moving them, they're going to move in pairs. So it's going to be 2 hydrogen atoms, which would be 2 protons and 2 electrons, right? How they're going to be moved is they're going to be added to an electron carrier. And this, again, this electron carrier molecule or molecules, this electron carrier molecule, this is NAD, and it's going to become NADH, which again should be familiar from Bio 1, but we'll talk about it and I'll show you a picture in a second. Or, as we'll see at the end of the Krebs cycle, or like citric acid cycle, this would be FADH to FADH2, with a subscript of 2. These are going to be the electron carriers. These are going to be the molecules that, because they're gaining hydrogen atoms, they're going to be moving from food, glucose, sugars, fats, proteins. Enzymes are going to remove those hydrogen atoms and stick them on these carriers, particularly this one. We're going to see a lot of NAD to NADH. Okay. So the NAD, the food item, is going to be oxidized. The glucose is going to be oxidized because it's going to be losing hydrogen atoms. And the NAD is going to be reduced because it's gaining hydrogen atoms. So this is the oxidized form of NAD. This is the reduced form of NAD, NADH. Yeah, that was a little easier than what I had in those PowerPoints. So I think we're just canned from the instructor. I mean, from the publisher. Sorry about that, Zoe. I was on the board, so I didn't see that you were waiting. I apologize, but it is being recorded, and I'll try to post it today or tomorrow. Okay, so now let's go back to me sharing this screen with you. I really don't want to draw NAD to NADH because that is quite a feat of strength. And I don't know, I just don't have it in me to that. So, blah, 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 this we talked about. So this is what NAD stands for, nicotinamide adenine dinucleotide. Don't worry about this respiratory chain character, it's not important. Here is the picture that I wanted to show you that I didn't want to draw, right? And I think you can see why. So if we take a brief look here, where we have, this is the oxidized form, nicotinamide adenine dinucleotide. So I'll just take a tiny second to run that through with you, right? So NAD stands for nicotinamide, which is a nucleotide. So it is a pyrimidine plus a sugar plus a phosphate. It's just a nucleotide that we don't use to make DNA or RNA. And then it's called nicotinamide adenine dinucleotide because there is also the nucleotide A. So we have regular old adenine, regular old purine adenine attached to a ribose, a monosaccharide, and a phosphate, right? So because there are two nucleotides, that's why it's called dinucleotide. So nicotinamide adenine dinucleotide. All right, so this is the oxidized version, reduced version, and here it says from substrate. You can think of it as from glucose because we're going to break down glucose and go through the whole process of glucose to carbon dioxide and water. And here you can see the hydrogens being added. We're going to add 2 electrons, but only 1 of the protons. So that's why the proton is out here as a plus sign. So when you see NAD depicted in one of these pathways, they're going to show it NADH, and that's it, right? But some textbooks will put a plus here, and some textbooks will also put a plus and H plus outside of that. So this is the most common electron carrier. FADH2 is exactly the same, except instead of nicotinamide up here, there's a flavin molecule, but it's also a dinucleotide. Now we should take a quick look at the molecule we're trying to build, which is ATP, right? That's going to come at the end of the electron transport chain, which is going to be the end of the complete oxidation of glucose. And again, most of you have seen this somewhere in your past. Same adenine, same purine, that were the same A with ribose and 3 phosphates. This is the same molecule we would use to make RNA. It's the same base, right? But it's attached RNA, it's attached to ribose, not deoxyribose. So here's adenine, here's the sugar, and then we have these 3 phosphates. And these little arrows here are supposed to help you remember that this is where the most energy is stored. When this bond is broken between the 2nd and 3rd phosphate, the most energy is released. There's still some energy released when you break the 2nd bond, but when the 3rd bond is hardly ever broken, because when you get to AMP, usually this phosphate goes up and hooks onto the adenine and it forms a circular molecule that we call cyclic AMP. All right, blah, blah, blah. So the pathways we are going to focus on for the exam, right? So you need to be able to name the step or pathway, and we'll go through them one at a time. or the complete oxidation of glucose, which is also known as cellular respiration. And there are 4 steps of that pathway, right? There's glycolysis, the prep step, the Krebs cycle or citric acid cycle, and the electron transport chain and chemiosmotic synthesis of ATP. Okay, so that's what I mean by name the step. You need to be able to know each of those 4 steps. You need to be able to name the starting and ending organic molecule for each step. You need to count and keep track of these molecules, ATP, NADH, CO2, FADH2, and you need to know where in the cell each happens for prokaryotes and eukaryotes. And again, this should 100% be reviewed from Bio 1. If not from Bio 1, then from Chem 2, from Chem 102. And if you didn't learn it in those places, it's, you're going to have to learn it now. Okay, so this is a quick summary of the same thing. And then after we talk about glycolysis, we will talk about aerobic respiration. But I'm going to skip ahead to this picture and use it for our discussion of glycolysis. See if I can just make that small. I can, and make that small, and make myself big again. Not to share, make me big. How do I do that? Sorry. Okay. All right. Okay. So I'm bring you here, and I'm going to erase. Looks like the blue one doesn't erase very well. Lucky for you, I have this. It's dry now, but it was right there. Where is it? Okay, so complete oxidation of glucose. And the reason we talk about glucose is because it is the most commonly digested monosaccharide in all of eukaryotic cells and prokaryotic cells. And this complete oxidation of glucose, this first portion, glycolysis, and then we have the prep step, right? And then we have the Krebs cycle. You may have learned that as citric acid cycle, or you may have learned that as the TCA cycle. And then the last portion, the electron transport chain plus ATP synthase. Right? Those are the 4 steps that I mentioned that you have to know the starting and ending molecule for each one, and you have to keep track of those molecules I listed and where they happen inside the cell. This portion, glycolysis, the prep step, and the Krebs cycle, this can happen with or without, or all can happen without the presence of oxygen. If we use the electron transport chain, and if the final acceptor, the final electron acceptor on the electron transport chain is oxygen, and only then do we call this aerobic respiration, because we used oxygen, right? If the final electron acceptor is oxygen, then we refer to this, you may hear this whole process referred to as aerobic respiration. Now, you've been sold a little bit of a bill of goods, right? Because we try to keep things super simple in Bio 1, and we don't give you all of the vocabulary, even though it seems like we do. Technically, only if you use oxygen as your final electron acceptor is it aerobic respiration. If you use something other, if the cell, when I say you, I mean the cell, the bacteria, uses something other than oxygen as its final electron acceptor, then we call it anaerobic respiration. And if the cell only does this, glycolysis, that's fermentation, okay? With the end products of glycolysis being turned into a little bit of ATP and a little bit of byproducts, organic acids or alcohol. All right, so we're going to start by going through these 4 steps relatively quickly, and I'm going to talk about the starting molecule, ending molecule, and what's produced, and basically just quickly what you need to know for the exam. Okay, so I'm going to erase everybody except glucose, or glycolysis rather, and I'm going to go through glycolysis again relatively quickly. I get it, but save up your questions, ask me at the end, or next time I see you, which will be Monday, which will be the day before the exam. Okay. All right, so glycolysis starts with glucose. That's the starting molecule. There are many enzymatic steps, so I'm just drawing many arrows. There's 8 of them all together with a couple that are interchangeable. And the end product is we produce 2 molecules. Double checking which one your book goes with. 2 molecules of pyruvic acid, old school. Pyruvic acid. Now don't freak out. If your book in chemistry or your book in Bio 1, I know for sure, called them 2-pyruvate, that just means that the hydrogen hasn't left yet, right? So, or has already left. With the acid form, that means there's a hydrogen sitting there that can leave and donate to the solution if the solution's in water, right? Pyruvic acid is a 3-carbon molecule. I'll show you a picture in a minute. And we have 2 of them. And glucose is C6H12O6. And if you haven't had to learn that yet, you should at least be able to associate glucose with that chemical formula. And you can see right away there's 6 carbons in glucose. So I have 6 carbons here and 6 carbons here. And since we release carbons in the form of carbon dioxide in the oxidation of glucose, you can see that we're not going to release any because we have 6 on this side, 6 on this side. So starting molecule, ending molecule, good, check. So name of the step, glycolysis. Starting molecule, ending molecule, check. Things we have to keep track of. Let's start with NADH. There are going to be 2 NADHs produced, and I'm just going to draw them on an arrow coming out of the cell, out of the chemical reactions. So we're going to make 2 NADH. And again, this could be written just like that. Go over here and go high. Some books are going to write it like this, NADH. Some books are going to write it NADH plus. Other books are going to write it like this, NADH plus and H plus. That all means the same thing. And I know it seems like a tiny detail right now, but I remember, at least for me, when I was in your shoes, I didn't understand why we called the same molecule 3 things. Depends on how chemistry oriented the person is, whoever it is, that is, sorry, I have to admit someone. Let's see if that works. No, not good. All right, so 2 NADH are produced. We already said 0 carbon dioxide are produced. And we are also going to make 2 ATP, but it's a little confusing because in glycolysis, you have to actually input 2 ATP to get the process started. I'm going to draw them in a happy yellow sun because they make energy. And you produce 4 but this is a big important but, you only net 2. So I'll say that again because I know it's a little confusing, right? So we input 2 to get it started. So we're going to have to add phosphates to some molecules. We actually produce 4, right? So that would be our gross. So we subtract the 2 that we put in from the 4 that we get. So we net 2 ATP. Okay, so that takes care of the carbon dioxide, the ATP, the NADH, and we produce 0 FADH2 at this step. So the last thing you need to know is where this happens in the cell. And I will just tell you, it happens in the cytoplasm of both bacteria, prokaryotes, for prokaryotes and eukaryotes. Done. Step one. Step two is known as the prep step, or at least that's what I'm going to call it. It has a more complex chemical name, but it's not important for me that you know that name at this point. The prep step is necessary to get us from glycolysis to the Krebs cycle, to the next, to step three. The prep step, the starting molecule is going to be pyruvic acid. And remember, we have two of them because glucose has 6 carbons, and we basically snapped it in half and rearranged it. So we have 2 pyruvic acids. This is a single arrow because it's one enzyme. Converts the pyruvic acid to this molecule. Acetyl coenzyme. Coenzyme A. Oof, long word, right? We usually just abbreviate to acetyl-CoA for coenzyme. Coenzyme, word we just refreshed, right? So it's a helper molecule to the first enzyme of the Krebs cycle. It's organic, but it is not a protein. It only has 2 carbons. 2 carbon molecule. Remember, pyruvic acid was a 3 carbon molecule. But just like we had 2 pyruvic acids, we produce 2 acetyl-CoAs. I have 4 carbons on the right-hand side. I still have 6 carbons over here, which should indicate to you, oh, something happened. We lost a carbon. I'll make those blue because I have this blue. So we're going to release 2 carbon dioxides. That's how we're going to get from 6 carbons on the left, 4 carbons on the right, to 3 carbon molecules. All right, so we have 6 over here, 4 over there, release 2 is CO2. We do not make any ATP at this step. We do produce an NADH for each pyruvic acid. I'll say that again. We produce 1 NADH for each pyruvic acid. We have 2 pyruvic acids, so I'm going to produce 2 NADH. And let's put those in a rectangle to help us find them. Okay. Where does this happen in the cell? Is different bacteria, cytoplasm. Cytoplasm for the bacteria. And for eukaryotes, this happens in the outer membrane, the outer phospholipid bilayer, the outer membrane of the mitochondria. All right, sadly, I can't keep this. I can't preserve this for us. I'll let it sit there for a couple seconds. You guys can take a screenshot of it if you want, so that you don't, it's not so hard to find if you're watching the whole lecture. But we have 2 more steps to put on here, and I am out of whiteboard, so I'm going to have to erase this, unfortunately, so that when you ask questions, we'll have to recreate it, I guess. So I'm going to just take my time on purpose erasing this. It's so beautiful. Maybe, I have no idea. Maybe we can just flip it around. Use the chalkboard for the last 2. How about that? Oh, yeah, let's do that. And then we can save it in case you have questions that you want to ask. Again, from our ones, organic molecules out there for them to practice. All right, so I'm just going to get out a piece of chalk instead, and we'll talk about the Krebs cycle, step 3. Goes by many names: Krebs cycle, citric acid cycle, CA, which does not stand for the citric acid cycle, even though it seems like it should. It stands for the tricarboxylic acid cycle. It's the only cycle we're going to talk about, so it's a circle. And again, we're going to talk about a starting molecule and an ending molecule, even though technically, because it's a circle, it doesn't really have a start in it, right? But I will give you molecules you can use for this discussion. The starting molecule is going to be two molecules. It's going to be an acetyl coenzyme A molecule that we produced by our prep step, joining with a 4-carbon molecule that's known as oxaloacetic acid. To form a 6-carbon molecule that is citrate or citric acid. That's why it's called the citric acid cycle. I know that's very small and you can't see it very well. It's all really large in your book, right? That's it as far as what you need to know for the starting / ending molecules. The starting molecules will be acetyl-CoA plus oxaloacetic acid to form citric acid. And technically then the ending molecule is going to be oxaloacetic acid because it's the last in the cycle. That's it. You don't have to know the names of any of these other ones. These are not very good arrows, but they're supposed to be arrows going around in the circle. What we do need to focus on is molecules that are produced along the way. Because remember, that's the second thing that you have to know. And I just need to get this as close to correct as I can. So I'm just bringing my book over a little bit. Because technically, different books show this happening in different places. So I'm just going to put it here. At this juncture, we get a carbon dioxide and an NADH. So I'm putting them in a circle and a square and keeping it the same colors. At this junction, we produce a second carbon dioxide and another NADH in the green rectangle. And down here at the very bottom, ooh, I have a yellow. We actually produce an ATP, which again, I'll put in a happy yellow sun because that's going to be our energy molecule the cells are going to use to do work. Here at this step, we produce a new molecule of FADH2, still an electron carrier. I'll put him in a triangle. And then I believe up here at the top, right before we produce the oxaloacetic acid, we make one more NADH. Oh, crap. Sorry. Put that in a square. Yay! Now, over here on the side, I often tell students to make a little chart for themselves. 1 turn of the citric acid cycle, 2 turns. And then I put the molecules running down the middle, carbon dioxide, NADH, ATP, and FADH2. They kind of come in that order. And then I go around and say, usually out loud to the class, but I'll just give you guys a second to think about it, right? If I turn the Krebs cycle 1 time, so if I put in a single acetyl-CoA, I'm going to produce, look for the green rectangles, 1, 2, 3, oh, sorry, the blue circles for the carbon dioxide. I'm going to make 2 carbon dioxide, 1, 2, 3 NADHs, 1 ATP, and 1 orange triangle, 1 FADH2. If I go around twice, you guys can easily see, I would get double of all of those. And why would I go twice? If I start with glucose, then it's going to be 2 turns of the PCA Krebs cycle. And that's important because in cells, you're not always going to start with glucose in the real world, right? In our little perfect example, You always start with glucose. We always get to pyruvic acids. We always get to acetyl-CoA, so we would go around twice. Where in the cell does this happen? If you are bacteria, still in the cytoplasm. Prokaryotes. If you are a eukaryote, it's going to happen in the matrix of the mitochondria. And yes, you have to know that for the test. All right. Now I'm going to bring you guys back and use the picture from the book for the electron. Why? Because I'm tired. Yellow is hard to see. Sure, I can try to use a yellow, a different color. I don't know. I'll have to go down to my collection and see if I what I have down there. I like the chalk too myself, Lauren, but I think some people don't like to see a chalkboard, but I like a chalkboard. All right, I need just a little mental pause here. This is long, and I'm out of practice. And I'm old now, so it's not as easy as when I was 30, right? All right, so there's a brief summary of the TCA cycle. Here it is with all of its little bits, and that's the same picture that's in your book now, maybe with some different color changes. Again, you don't need to know the names of all of the intermediates, and please don't ask me if you need to know the names of all the enzymes. They're not even on this picture, okay? You need to know the things that we went over. Now, the electron transport chain is another story altogether, okay? That's a little more, we need to focus on that a little more in detail. This is the picture. I know it's super complicated. Relax. We're going to look at it one step at a time. This is the fourth step, okay? And there are some words that are going to go with this. I'm going to say the words while we're looking at the picture, and then I'll make you a slide that has just the words on it, okay? So this is depicted, or the picture that they're showing you is the electron transport chain in a gram-negative cell, okay? So remember, the gram-negative cells have that outer membrane of phospholipid bilayer, and then a few layers of peptidoglycan, which they are not showing, and then the cell membrane. Oh, look, actually it says cristae. So this is actually in a eukaryote, but I'm pretending it's a gram-negative cell. If it was in your cells, this would be the outer membrane of the mitochondria. That's what fooled me, because it just said outer membrane. And the crista, or the cristae, are those folds of the inner membrane of the mitochondria. In bacteria, this is the cell membrane. Remember, we said one of the functions of the cell membrane on Monday is that it is the site or location for metabolism, for the electron transport chain. This is what we were talking about. So see all these different colored things? They are protein, they're groups of proteins. You don't have to know their names, it's not important. There are actually 9 of them or more. So when I write it out for you, I'm going to say 9 or more proteins in a membrane, because it's various depending on if it's plants do this too. And they do this for photosynthesis as well as for cellular respiration. And different bacteria may only have 8 of these, or they might have 12 of them. So the number is different. So I'm going to say 9 or more proteins in a membrane. And I'm leaving it purposefully general, because this could be the mitochondria membrane, or it could be the cell membrane in a bacteria. So we have these proteins in a membrane. What do? They pass electrons. All the proteins in the membrane, each one of them is a little more hungry for electrons, or a little more electronegative than the protein in front of it. So when the electrons get here to the electron transport chain, the starting molecule is our old friend NADH. It's bringing all of the NADHs that we made in glycolysis, the prep step, and the Krebs cycle. All of them come to the electron transport chain, and they are the source of the electrons and the protons. So those hydrogens that we took off of glucose, we stuck onto NADH to carry around. Now we're going to rip them off. The electrons are going to go one way. See the black lines? They get passed from purple to orange to green to pink to darker red, orange, pumpkin color. And then the electrons are going to be given at last to oxygen. Oxygen is the final electron acceptor on the electron transport chain. There's oxygen. It's going to grab these extra electrons. And what we're going to see is that is dangerous, but it's not going to last for very long in the cell because protons are going to be being pumped through ATP synthase, and they're going to hook up with that oxygen with the spare electrons and form water, which we know is cells need it. It's wonderful. It's harmless. Okay, so yes, you already have access to this PowerPoint and the Zoom. Well, the Zoom after it's over and I post it, but yes, yes. So, and it's in your book. It's the same picture that's in your book. So the hydrogens, the protons, where are they coming from? They're also coming from NADH. They get ripped off. Those are the protons that got put on from the glucose when we took the hydrogens off to oxidize the glucose. And not all of the proteins actually pump protons, just some. So all of the proteins pass electrons. Some pump protons. This guy, this guy, this guy. They pump them from one side of the membrane to the other. There's more of them outside than inside. They can only get back through the membrane through ATP synthase, ASE. That means it's an enzyme. That means it's a protein. What does ATP synthase do? Its active site grabs ADP and puts a phosphate on it to become ATP. See the happy yellow sun? But, this is really important, but it can only do that with a fuel source of protons, of positive charges. It's just how it works. So we have to have the protons to make ATP synthase produce the ATP. Most of the protons are going to be recycled, but 2 of them are going to join with each oxygen to form water. Okay, so when this is working at top speed, it's also a circle. It's going on all the time in your cells and in bacterial cells. And for every NADH that we bring to the electron transport chain, that's enough protons to produce 3 ATP. Every FADH2, which I made in a triangle, but they've got in an elliptical an ellipse here, right? FADH2 doesn't bring its protons and electrons to the first molecule of the electron transport chain. So we miss this first pumping of protons. See how that's before the purple one? And we're getting purple dropping off at purple here. So there are fewer protons, less fuel, less ATP. FADH2, lower potential energy. It's like the fancy chemistry way to say it. Lower potential energy only brings fewer protons and fewer electrons to the electron transport chain. So we only get 2 ATP for every FADH2 that comes to the electron transport chain. We only get 2. If we had glucose and we are in a bacteria, then we actually get all 38 ATP from glycolysis, the prep step, the Krebs cycle, and the electron transport chain. Most textbooks will give you the eukaryotic numbers, which are between 32 and 34, 36, depending on the cell and depending on how efficient the electron transport chain is. If it is anaerobic respiration, so if we don't have oxygen there as that final electron acceptor, there's no oxygen here, if there's something else sitting here, we don't make as many, usually also that involves not bringing as many hydrogens, we don't make as much ATP. For anaerobic respiration, that's mostly in bacteria, those cells produce somewhere in the 20s of ATP per glucose. And the reason I can't give you an exact number is it's different for different organisms, and it's different depending on their current chemical situation. If their system's working at maximum capacity, or if it's somewhat inhibited for whatever reason, not enough time, not enough heat, no, too hot, not enough water, whatever. Fermentation, let's take a look at fermentation real fast. I think this guy. So this just goes through the anaerobic respiration. And for fermentation, we mostly have two options. We're almost done, guys. Hang on for like another 10 minutes. For fermentation, we show you these two options, partly because they're very common and partly because we think they're ones that you are familiar with or know a little bit about. This fermentation process where we have glucose, so notice there's no electron transport chain here and there's no Krebs cycle, which is where you generate most of your NADH. So we have glycolysis, and then we have one more chemical reaction to form lactic acid. This is what's happening in your muscle cells when they form lactic acid or lactate, where you feel a burn in your muscle when you're lifting weights or you're running or whatever, because you're out of oxygen to do aerobic respiration. So some textbooks will say you switch to anaerobic respiration, but technically you're actually switching to fermentation. And bacteria and yeast, they give you the yeast example here because that's more commonly known for people as far as yeasts are important for making bread and making the gas that makes the bread rise and for making alcohol, right? So in the yeast system, we would have glycolysis, again, no prep step, no Krebs cycle, no electron transport chain. We have a couple of chemical reactions here, a couple of enzymatic steps. One releases CO2, that's where the bubbles come from, and they produce ethyl alcohol as a byproduct. So think about if you make wine, that carbon dioxide has to be released, or even beer. If you make it at home in a big vat, you have to have a regulator at the top that will release the carbon dioxide, or the pressure can build and cause an explosion, which would be bad because there's a lot to clean up, right? Bacteria can make all these different kinds of acids. They took this picture out of the current edition, but I love it because it shows you all the different possibilities from pyruvate from a bacterial standpoint. It's not just about making ethanol or lactic acid. There's lots of other possibilities. Why do you care? Because we base the biochemical tests for these particular bacteria on these end product acids. We can put in a chemical indicator that will tell us, do they make this acid? Do they make this acid or that acid in particular? And we can use it to identify a particular bacteria. And then the very last picture in the chapter is this one to remind you that the anabolic and catabolic processes are linked. You're going to build bigger polymer organic molecules and structure from the monomers, as well as you can break any of those things down and put them into this central pathway that we just talked about, right? Glycolysis, pyruvic acid, acetyl-CoA, TCA. And you can see the connections here between the other organic molecules, proteins, lipids, and even nucleic acids, although we usually break those down last, okay, especially as a source of ATP. However, when there is excess, notice the purple lines coming out. When there's excess, the cell's going to build fat or fatty acids, or the cell's going to build amino acids that can then be converted to nucleotides to make more DNA and RNA. All right, I'm going to stop the share and end the recording.